little while back I did a video on bringing the woolly mammoth back, and that brought up the topic of CRISPR, and I thought it sounded really interesting, but I wanted to look into it. But I wanted to make sure I actually get an in-depth look, so this is that video. Let's start off slow and get a very high-level overview of this topic. What is CRISPR and how does it work? The ultimate guide to CRISPR mechanics, applications, methods, and more. First of all, CRISPR stands for clustered, regularly interspaced, short palindromic repeats. It is a part of a bacterial immune system that's DNA and has been repurposed as a gene editing tool. It acts as a precise pair of molecular scissors that can cut a target sequence of DNA that is directed by a customizable guide. It's important because it allows scientists to rewrite the genetic code in almost any organism. It was discovered by these two, Dr. Jennifer Doudna over here and Dr. Emmanuel Charpentier over here. This paper proved that it could be repurposed as a gene editing tool. Let's go ahead and tuck that away for later. Oh, interesting. They won the Nobel Prize in Chemistry for this discovery. The first all-female team to do so. Applications of CRISPR that could happen. Cell and gene therapies. Might be able to cure a range of genetic diseases. Could be used for diagnostics to kind of get the lay of the land for, it looks like it was even used for coronavirus. Gene editing for agriculture. Create crops that are disease resistant and drought resistant. Bioenergy. Okay, I feel like this article is pretty well laid out. And if you would like to just read this article, it will probably give you a pretty good understanding. But there's a lot of stuff they're going into that I'm not understanding right off the bat. And I feel like it might be worthwhile to make some side trips and understand some foundational things about CRISPR and like how it works, and then come back to this and see if I can explain it a little more thoroughly. So I had an idea. I'm just going to ask Betsy, what things would I need to understand first before I would be able to understand CRISPR? Okay, let's look at these things individually, see what they mean, and then go from there. Starting with DNA structure and functions. I mean, I already have a pretty basic understanding of DNA structure and functions from when I was in high school, or just like casual learning through life, but let's make sure I know what I'm talking about. The structure and function of DNA. Okay, I'm gonna read this article and find out what it says, and probably read a lot of other articles and find out what they say, and then I'll be back to tell you what I learned. I'm back, and I'm gonna be real honest with you guys. It was a struggle, but let me get into it. So Betsy told me to look into DNA structure and function, right? With the nucleotides, base pairs, and double helix. Wow, I had no idea what this was when I wrote it down. And now I do, which is kind of crazy. So I started looking into this article. I think this is the one I was reading on camera. So I found out that DNA stands for deoxyribonucleic acid is what DNA stands for. I haven't found out exactly what deoxyribonucleic means. I found out a little bit about the acid, but we'll talk more about that later. So before, they thought that DNA was just a polymer, which a polymer is basically just a long strand of things where there are like similar or identical pieces and they're all repeating in a pattern. So it's like 1, 2, 3, 1, 2, 3, 1, 2, 3, and they're in a strand. So they thought it was just a single-stranded polymer, but then they found out that it was the classic double helix strand. I don't know if they found out that it was double helix yet, but they found out that it was 2 strands, not just 1. I don't know why I linked this word to this article because I can't find it in here. Anyways, I was having a really hard time understanding what was going on in here. And now that I do understand more, I kind of want to go back and read this because I'm looking at it and I'm understanding a lot more, but that's not what happened. So I was really struggling. So I was like, okay, let me just focus on, forget about Betsy and her suggestions of what might be a good way to do. I'm just going to think about how DNA is read by the cell, because I know that DNA's got this code that is like a sentence, but I want to know how the heck that information goes from being like a code in a DNA strand to like producing something. I don't even know what it produces yet. So I looked up how is DNA read by the cell, and that brought me to this article. And I learned that you can think about the protein coding sequence in a DNA strand as kind of like letters in a sentence. So the sun was hot, but the old man did not get his hat. So if you were to take the sentence and split up all of the individual 3-letter words, it would make perfect sense. Like you can kind of read it like this, but you can also split it up into this, which makes a lot more sense. But if you were to like kind of shift the reading frame by like 1 letter, it would stop making as much sense. Or if you went the other way, there's only one way to read the DNA code, the gene codes. This was a really interesting article, so very helpful. Also, this graphic, I didn't know this until later, but this graphic is used to look up what the stuff is going to be in a DNA strand, like what 3-letter pair it will actually be, and it's really interesting. We might get back into this later, but this is a cool little graph. So I'm still not sure what exactly is reading these little sentences at this point. So I tried to search this, what is reading DNA, and that didn't come up with anything. So I changed it. What part of the body is reading DNA? And that brought me to this article here, DNA basics. So the 4 chemical building blocks that DNA is made up of are called the nucleotide bases. These are their actual names, but you usually just call them A, T, G, or C. In their little pairs, they're always going to be opposite their letter pair. So T is always going to be opposite A, even if it's on the flip side, and C is always going to be opposite G, even if they're on the flip side. So A will never go with G, and T will never go with C. And the backbone of the DNA helix is, this little blue part is a sugar phosphate backbone, which will probably come up later, but I'm not sure how it applies yet. So because they're complementary, if you know what one side of the strand says, you will automatically know what the other side says, just because you know that the pairs always go together and they can never go with another letter. So in this DNA strand, you have the genes, which I had never known this before, but I found out, which is a segment of DNA that provides instructions for making a specific protein. But it's only this one little part of the DNA strand is the gene. And then inside of that gene, so it's like this specific sequence of letters is the gene. And over here, there's a different specific sequence of letters, but in the gene, there are special coding sequences which can code for things like proteins, like specific proteins, like the alcohol dehydrogenase protein. So we know that the gene is coding. So we know that this DNA strand holds little sentence codes, and we know that the gene is the one that holds the specific sentence code that will tell a protein, or like talk to the cell and tell it what to make. But I'm not sure how yet. So at this point, I still don't know what a protein is, really. And I don't really know what deoxyribonucleic means, or like why it's called an acid. Why is it called an acid? So there's still a lot I don't know, and I'm trying to figure it out. First of all, like, how does DNA transmit information to the cell is what I am like thinking, maybe this is what I need to know next. How is that sentence code getting transmitted to the cell, and then things are getting done, accomplished? Which brought me here. The information in DNA is decoded by transcription. Okay, so it's a 2-step process. The first part is transcription, and then translation comes after that. But they keep referencing the cell. What are they talking about when they say the cell? Because it's like, where is the DNA actually? It's inside of a cell. So are they referencing like that specific cell that the DNA is in, or is it affecting other cells? I just know very little at this point. So I needed to know where we can find DNA in the first place. So I came here, where is DNA found? And it turns out that yes. So DNA is found in the nucleus of the cell. Tiny little cell here, and inside this nucleus, and the DNA is found in these chromosomes, which I never really knew what chromosomes were. Who knew? Did I even take high school biology? The strands of DNA are coiled up super tight, and in those coils are wrapping around the chromosomes. I think there's something inside of here that's giving it structure, but that's where the DNA is. So at this point, I was pretty stuck. I also just didn't have very much time to look into it, so my wife, who is a super big biology nerd, offered to help me out, which I was very grateful for. So this is what we talked about. She explained some things. So first of all, there's something called the helicase that I don't know where it comes from. I'm pretty sure it's a protein that hits up the DNA strand and breaks it in half. So it turns it from DNA into RNA, which is a single strand. So RNA is single-stranded, some like sentence coding, and DNA is the double-stranded helix. So the helicase breaks it up into 2 strands, and there's something called an RNA polymerase. I'm explaining this very poorly. Okay, this is a better picture. Okay, so here we've got the helicase breaking up the DNA into 2 strands, and then the DNA polymerase, which is these little yellow things, come in and start to pair it with mRNA, which is messenger RNA. And because, why are there these free nucleotides just hanging out here in space? I'm not sure. So because we know what the other half of the strand says, because we know what this whole single strand says, it can pair it with another one and kind of like transcribe it. So this is the transcription part, where pulling these free nucleotides from where though? I think they're kind of just hanging out in the cell, maybe? So it's pulling these free nucleotides in and putting them here and pairing them, and this is the messenger RNA, the mRNA. And for some reason, in the RNA, there's no T's. It only has U's. I don't understand why, but that's what she said. So fun fact about DNA and RNA, DNA can only be in the nucleus of the cell, but RNA can be in the nucleus and the cytoplasm. What's the cytoplasm, you ask? I'm not sure yet, but just so you know. So this DNA polymerase is actively building another mRNA strand. It's using this initial code, this initial DNA helix code as a cheat sheet to form the next strand. So then a protein,
Okay, so then a protein, I don't see it here, but a protein grabs this strand of mRNA, this yellow thing right here, and pulls it out of the nucleus and puts it over here into this ribosome. What's a ribosome, you ask? I'm pretty sure it's just another protein, but I'm not sure yet. So anyways, so the mRNA with these little copied, this transcribed little letters, sentence code, gets fed into the ribosome, and as it does, the tRNA over here, these little weird looking guys, have their code letter. It's three, they only ever have three at a time. So they get pulled into the ribosome, and when they do, they match up with the mRNA over here. So this one we know is going to pair with that one over there because G only ever goes with C, C only ever goes with G, and A goes with U, for some reason not T. I don't understand why. And then once they're here inside of the ribosome, there's a something. It forms like a polypeptide bond or something. So they're bonded together, and then the strand gets moved out of the ribosome, and as it does, this free tRNA thing goes whoo, off into the void, and it finds another amino acid, and then this chain grows longer and longer and becomes a polypeptide chain, I guess, because they're polypeptide bonds linking them together, or something like that. So apparently there's just a bunch of tRNA cells with amino acids bonded to them just hanging out in the cell, and that's where these are coming from. And there's like a strong or like a weak something force, chemistry force, pulling them together, kind of like magnets, and that's how they know. So they're just hanging out here, and as the mRNA strand comes along, they just get pulled into the right spot because they know which one is which, because they're bonded that way. So, oh, sorry, I forgot to mention that each of these three letter pairs, because they all only have three, are called a codon. So these three little pairs on the strand are called codons. And if you go back to, let's see, if you look at these codon chart wheels, they'll tell you if you got this letter plus this letter plus this letter, that'll be this thingamajig. And if you got this letter plus this letter plus this letter, it'll be this one. So they're very specific things that will code for different proteins later on. But you need to know which one is which if you're going to make the correct protein. But I haven't found that out yet. We're still learning about that. Anyways, and this whole process right here is called translation. So it's transcription when it's in the actual DNA, and this strand is copying off the cheat sheet of the initial one. And then once this thing is gone, the DNA will kind of just form itself back together. It'll get pulled back together because the individual base pairs like each other, so they will just pull themselves back together once this thing is gone. And then this process is called translation because it's translating the like DNA or the mRNA language into the tRNA language and turning it into amino acid language. It's translating it. So there's something called methionine, which is the amino acid over here that will always start the process. So the first one is always methionine. That's always the first one. And that's pretty much all I have so far. So I'm going to keep looking into, I think I know like a fair amount enough about like DNA and how it's working. I think I need to start looking into like I know there's a process in the CRISPR thing that's like hijacking like the defense mechanism of the cell, and it's using it to do things to the DNA. I think I need to learn more about that process. So I'll be back with those sweet deets later. It's sweet deet o'clock. So what we should probably start with is looking into the immune system thing that is doing the actual code rewriting, because I heard that was when I was doing the woolly mammoth thing. Read something about that. So that's probably the next step, or I should find out what a protein is, because I feel like that's relevant. So that's what I did. I started looking into proteins, and that brought me here. What are proteins and what do they do? First thing I learned is that proteins are complex molecules that play many critical roles in the body. They're sometimes referred to as the workhorse of the cell, I think. It doesn't say it in this article. But proteins are made up of those amino acids, hundreds or thousands of smaller units of amino acids, all attached together in a long chain. There's only 20 different types of amino acids, which I thought was a surprisingly small number. But out of those 20, they can be combined into like millions of different combinations to make all of the different proteins that need to be made. These are some of the types of actual proteins, things like antibodies, enzymes, messenger things that will carry signals throughout the cell. They can be structural components of the cell. They can transport things in the cell or store them. So proteins are kind of just like basically anytime there is something like actively doing something in a cell, it's probably a protein doing it. Which I feel like anytime I had ever heard people talking about proteins in the cell before, I was always like, oh, but I had no idea what they were talking about. But now I do. So after I looked at this article, I was still kind of curious, is everything in the cell a protein? So I searched that up and it brought me here. What are proteins? And this is where it talks about proteins being the workhorse of our bodies. So not everything in the cell is a protein, but there are a lot of proteins in the cell doing stuff. I don't know if I talked about this before, but proteins, like the reason we eat protein, like milk and eggs and meat and whatever, is because your body digests that, breaks them down into their own individual amino acids, and then those amino acids can be used to make the proteins in the cells. And that's why it's important to have proteins. Who knew? All right, so then I wanted to know a little bit more about like specifics. So what are some examples of proteins? Because I don't really know what all these things do, antibodies, enzymes, whatever. I don't really have a good foundation in what these proteins do. So this gives me more of kind of similar information. Proteins do things like they start chemical reactions, they hold things together in the cell, they regulate things going across cell membranes, they can like guide things. They'll be like the air traffic controller people with the two lights guiding things around in the cell. So one thing I did learn that's pretty crazy is that proteins strands of amino acids can be 574 chains long, which is a lot. And if even one of them is off, it can result in bad things like sickle cell anemia for one, which is pretty crazy. So you best get your protein amino acid chains correct. Okay, so at this point I needed to do a little recap in my brain, so I was understanding what was going on. So, all right, at this point I still didn't really know how amino acids are actually turned into proteins. Like you've got this long string of amino acids. How does that magically become another shape that can do cool things? Also, as an aside, I heard when I was talking to my wife about this, she said that there's kind of a chicken and an egg problem, because if you've got your DNA with the code to string together some amino acids, or if you've got your DNA strand that has the information that proteins need to build more proteins, how do you get it to a place where the proteins can be built without a protein to help it along? So it's great if you already have proteins running around the cell building things, but if you don't have any proteins and you need to make the first one, how does that happen? So apparently this is a problem. So this is the next thing I looked into. How are polypeptide amino acid strands turned into proteins? And it brought me here. So it seems like the strands of the protein kind of like weave themselves together into a braid. So they're kind of like braided and twisted and folded to make a complex three-dimensional shape, but I'm not sure exactly how that happens. So I went here to look for that answer. And this one says, the folding process can be thought of as a drunken walk across an energy landscape, with the movements driven by intermolecular interactions and collisions, with the goal being to find the lowest point in the landscape, which I don't really feel like is a extremely revealing answer, but that is, it's enough of an answer for me, I suppose. While looking for this, I stumbled across mutations, and this is wild, y'all. We understand how DNA works. It's got these long strings of letters that can be turned into like sentence information codes, right? But what happens if something happens in that code and the letters get messed up? So if you have an original sentence, let's pretend that this sentence is what the DNA is saying. Quick brown fox jumped over the lazy dog. But if you have one letter off, quick brown fog jumped over the lazy dog. We got a G instead of an X in there. And now the sentence is still intelligible, but it means something slightly different. And voila, you have a mutation. So this one is fairly benign, but pretty soon you get ones that are pretty bad. So things like if you put a single letter in, like this letter G gets put in before the jump, you can see that the whole thing gets shifted over and you still have the same like grouping of the letters, but now things are really getting messed up. This word, dove, is still intelligible, but that's the wrong word. So you still sometimes get words, but they're not the right ones, or coded meaning. And then you get ones like deletion, where quick brown fox, the letter X was deleted out, but the J there still had to be a letter there, so the J is shifted over, and now this whole thing is messed up. And then on and on it goes with some other ones like inversion, where this is flipped around, or duplication, where you have the same sentence twice, or translocation, where just a whole section of DNA is just cut out and pasted into a different one, which I'm still not sure exactly how that works. But yeah, crazy wild. So the reason that things get mutated is because copying errors occur in the DNA if it is damaged by radiation or chemical toxins. So this is what causes mutations to happen. If the DNA itself gets damaged, then it can read things wrong or copy things wrong, and then it's real bad. And then you get DNA that is telling proteins to make other proteins that are not the right proteins. And then those proteins doing crazy shabusiness. All right, and at this point, because I don't really understand how like DNA is getting cut and paste, I was like, how is DNA getting cut in half? So I searched, is there a protein that cuts DNA in half to have a new gene inserted? Because it was talking about like that mutation where DNA just gets cut and pasted into the wrong spot. And I was like, how does that happen? And wouldn't you know it, That brought us to the money question. So the CRISPR search begins in earnest at this point because we have stumbled onto our topic question, molecular scissors made of enzyme and RNA. So this is not the only one that cuts things, but the CRISPR Cas9 molecule is what is cutting and pasting things in the cell. No pasting, actually, just cutting. But we'll get there. So apparently before CRISPR was a thing, there were other gene editing methods that were being tried and I think they were working. I didn't get time to look into those, but so the way that CRISPR was discovered is somebody noticed that there were a large number of these specific palindromic repeats in the DNA strand. I'm trying to remember where it says that. I'm not sure, but it definitely says it here. Okay, so you've got these palindromes in the DNA sequence. So it's not like if you have the palindrome taco cat or civic or tenant or whatever other palindromes, it's like the single word is being read forwards and backwards, but in DNA it's not quite the same. You've got like this GTT all the way GAC, and that's going one direction, but because the DNA strands are complementary, it's reading exactly the same on the other side in the opposite direction. So it's kind of like a normal palindrome, but it's a little bit different. It's a DNA palindrome. Anyway, so you've got a bunch of these palindromic repeat sections in the DNA strands. This site was a little bit crazy. If you have time, I would recommend checking it out because it just was like so many more questions reading this. So here's an example of another palindrome. So these palindromic sequences are being used as recognition sequences for the proteins that are reading things. So if a protein is going along the cell and it sees one of these sequences, it knows where it's at. It's kind of like orienting itself in the DNA strand. And then they know at which point they need to cut the DNA molecule. I don't understand how this word molecule is being referenced to the DNA, but I'm pretty sure this is the same as a DNA strand. A double helix? I wasn't able to find a good answer for that though. Usually these sequences of palindrome sections are around 20 base pairs long. They can be shorter, but they're usually around 20. And this is the CRISPR region. Like we learned before, CRISPR stands for clustered, regularly interspaced, short palindromic repeats. In the DNA, there are these short palindrome sequences that are interspaced and they repeat. So that's the CRISPR region. So saying CRISPR technology is kind of a misnomer because they're just talking about that region in the DNA strand that is repeating these palindromes. The important thing is the CRISPR Cas9, but we'll get there. Just as an aside, I read somewhere that the zinc finger nucleus was one of the previous methods of gene editing, but I didn't get a chance to look into it. Oh, I did not realize that this is what Cas stands for. So Cas is going to come up a lot, and that stands for CRISPR-associated genes. So right next to the palindromes in the sequence are these other genes that will tell the Cas proteins to cut things. So these CRISPR sections are kind of acting like a library of something that we'll learn about in a second. But once the Cas proteins are reading the DNA strand and they see these palindromic sequences, and then right after that, they see some other stuff, they can read that, and that will tell them where to cut later on in the DNA. So I came back here. So this article was really quite helpful. So that was the first one, the palindromes, and then I went to this one, functions within the cell. And this is where things get really interesting. So all of the stuff that the people are discovering when they're talking about like the CRISPR and Cas9 stuff was all discovered in a bacterial cell. So bacteria are, like we always think about them as like being the little parts in dirt that make us sick or whatever, but bacteria themselves can also get sick, and the way they get sick is they have a virus, this little guy right here. Let's find a better picture. Whatever, we'll just go with this one. Anyway, so we got the virus here, and the way viruses stay alive is they inject their DNA into other things, and then those other things that they've injected their DNA to start replicating the viruses in themselves, and then boom, dead cell, happy viruses. So that's what this virus right here is doing. It's injecting its DNA into this bacteria cell right here. Sometimes these viruses are called phages. So that's a bummer for the bacteria, but bacteria have developed a way to be smart about this. And what they do is when the virus injects its DNA into the cell, the bacteria will grab a copy of it and put it into this CRISPR section. A short sequence is inserted between the CRISPR sequence of the bacterial DNA. So now the bacteria has all of the different little pathogen DNAs in its CRISPR section, and it can use this as kind of like a library about all of the things in the past that have tried to hurt it. So when the CRISPR protein comes along, it can read that thing, and then an RNA molecule is produced based on that section of pathogenic DNA that's been inserted. That tells the Cas9 protein where to go and snip in the DNA. The green spaces are the palindromic repeats, and then here the spacers are where the virus DNA get inserted. So the Cas9 comes along, reads this stuff, and then there is tracer RNA, this yellow stuff that gets bound to the Cas9, kind of tells it what to look for, and then the Cas9 protein, or the Cas protein, go and look at the viral DNA, and then they're like, hey, this code matches that other code we had before in our library of pathogen codes. This is not a good thing. I'm recognizing it. I'm going to cut it up. So they split it in half, and then they chop it in half. So that way, if the DNA is cut in half, it's not going to be able to reproduce itself. It's not going to be able to tell any other proteins to reproduce that specific DNA sequence, which is wild. That was as much research as I had gotten done. And then I was planning to take another day in research, but then I slept in and my whole schedule got thrown off. So I wasn't able to do the rest of the part of the research that I was hoping to do. So I think what I'm going to do is I'm going to look at this cool interactive thing that my wife sent me, because she's learning to be a teacher, and she's got access to these cool resources where you can interact with things. And then after we do a quick little look at the interactive, I want to look into how CRISPR is being, or like how the CRISPR Cas9 is being used today. Like I heard there were some crops that are being engineered to be drought resistant. And we also looked at the mammoth, bringing back the woolly mammoth, and how they're using the CRISPR Cas9 to like engineer some mice to have hair. So I want to see if we can look into that. I might not have time, but okay. So here we've got our cool little interactive. So here we've got our DNA strand, and here comes the CRISPR Cas9 protein. So the reason it's called, I'm not sure why it's called Cas9, but there are different types of Cas proteins. This right here is a Cas protein, and there are different types of them, and the Cas9 one is the most versatile one. It's like super good at targeting and recognizing all kinds of different stuff. Other ones are a little less capable of seeing pathogenic stuff in their DNA strands reliably, but the Cas9 one is real good at it. So that's why we use the Cas9. Anyways, so you've got your DNA strand, here's the Cas9. Inside of here is the guide RNA, which I didn't really get a chance to talk about, but the guide RNA is basically anchoring the other RNA molecule into the Cas protein. So it's looking along the cell. It's just being, I'm not sure exactly why it's looking at the cell. I think it's just kind of gets pulled in somehow by something. Anyway, so it's looking along. It finds this section of DNA it's looking for because it matches the guide RNA. And so it knows it's got this guide RNA, and that is telling it what sequence to look for. So when it finds that sequence of DNA that matches the guide RNA, then it knows this is where I need to be. So it hunkers down, it splits open that DNA, gets up in there, and then it chops it right in half. And then here's the break in the DNA, and then the DNA gets repaired. I'm not sure exactly what's going on with these nucleotide pairs, like these base pairs. Why are they all, I think there's some damage when it gets broken. The DNA pulls itself back together because it wants to be together. I was told that a lot of the time it just leaves it floating like this, so there's just a split thing of DNA, which is kind of crazy. There's a lot more to this interactive. You might have the ability to look at it yourself if you type in this link. I'm probably not going to link it in the description. Anyways, let's look into how CRISPR is being used. Oh, also I was looking at this article right here, and it sounds like there are problems with this method. Still many potential challenges, but we'll get there later. Okay, let's look up how CRISPR-Cas9 is being used to engineer drought-resistant crops. Genetic engineering for crop improvement, drought tolerance, yield, and domestication. So this isn't actually talking about results. It's talking about the principle. Is this hypothetical? Enhancing crop resilience. Current review provides a general understanding of principles as well as optimization. When were these published? 2 years ago. I know that one of the women who kind of pioneered the CRISPR technology gave a TED talk on this 6 years ago, so it's been around for a while. Application for editing, stress management, crop towards enhanced tolerance has great potential. Wait, is this not actually being used yet? Have they not actually found out how to do this yet? They're just trying to do it still? 5 ways gene editing is making crops resilient. I don't care about this. Roles of CRISPR to mitigate drought. Okay, my brain is tired. Maybe if you're still curious, you can look into it a little bit more yourself. I am done, I think. So thanks for hanging out and watching. I will catch you some other time. Peace.